Okay, hello everybody. This is your first laboratory for VMED 5130, Veterinary Bacteriology and Mycology. I'm Dr. Hawke. I'll be the lab coordinator. Kim Legault will be your lab instructor. And we'll be doing some demonstrations for you today as to how to go about performing these lab exercises online. I know that sounds a little weird, but that's what we're going to try to do. So I just want to remind you that you have 3 handouts that we gave you in class: your laboratory guide, which you'll have to use to generate reports that you'll be sending in to us. Also, we have the Louisiana Manual, which is critical to understanding what we're talking about in these labs. Gives you all the descriptions of the procedures and tests, and then the appendix to the Louisiana Manual, which is referred to several times as you go through the lab exercise. So we're going to be assigning groups to work in the lab, groups of 4, but we're not really sure exactly how we're going to do that yet. We may do it by random selection. We may do it by alphabetical order. We're not sure exactly yet how we're going to do that, but you will be working in groups of 4. And we're going to start today talking about the first exercise, which we refer to as unknown number 1, and that's your saprophytic fungi and dermatophytic fungi. So your pathogenic fungi and your saprophytes. What we do with each of these lab procedures is we will give you a list of a number of different organisms that your unknown may potentially be. You're going to get either trichophyton, Microsporum canis, Microsporum gypsum, Aspergillus niger, Pacillomyces, Mucor, or Rhizopus. Now your trichophyton, your Microsporum species will be your dermatophytes. The others are your saprophytes. You'll receive 3 of these unknown cultures, and you will then have to observe their growth on stock agar, which we have an example here of a fungal colony growing on stock agar. Now, we don't handle the fungi like we do the bacteria. You don't streak the plates with the organism that you're subculturing to. So we're going to use this agar plate as an example of how you would transfer it. We're going to use a medium called DTM, which is a selective medium, which you'll have photographs of in your Moodle PowerPoints. So step 1 is to sterilize your loop, and we don't want to just hold it in the burner there until it gets super hot, because that would kill the fungus when you pick it up. So we want to dip your crooked needle here, your little tool that we've made to pick it up, and you're going to pass that into the flame until it catches on fire. And when that burns off, then you're able to do your transfer. So to do that, we're going to reach into the tube and we're going to pick up a plug of that agar that contains some of the fungus on it. Easier said than done. And then we're going to place that right in the center of the plate. So you just inoculate it into one spot, and then that mycelium is going to grow out from that area. So that's the way you inoculate any kind of plated medium with your fungi. Your next step is going to be to take another portion of that colony and flame that off just a little bit to sterilize it. Then we're going to take some cotton blue our lactophenol cotton blue stain, place that on a slide, just a drop of it, and we'll give you a close-up of this in just a moment so you'll see exactly what we're doing. But you're going to take some of that material again, dip it in alcohol, flame it off, and then you're going to go in and pick up another bit of the fungus, and you want to get more fungus this time than agar, and you're going to place it in that cotton blue. And you'll take the other probe and you're going to tease that back and forth on the slide until you see the individual hyphae being spread out where you can look at these under the microscope. So once you've made that slide, you'll be looking at it under the microscope, and you'll be looking for reproductive structures, which we will have depicted for you in still shots on Moodle. So for inoculation of DTM agar, we're going to be going into a tube that contains a culture of fungi here, and Kim will be picking up a little tiny amount of that on her probe that she sterilized, and then that'll be placed right in the center of the plate. So the organism will grow as a mycelium, or a large colony that's going to spread out on that plate, and it's going to produce a color reaction. If it's a dermatophyte, it'll turn the agar a reddish orange color that will be diffusing out from the colony. And we'll have illustrations of this, photographs of this on your Moodle site.